Hello students, welcome to your second session of chapter number 11 of your second PUC NCERT, that is Biotechnology Principles and Processes. In last class we had discussed regarding the definition of biotechnology. We had also covered steps involved in recombinant DNA technology, along with which we have also studied about construction of first recombinant DNA. Now in this class we will be continuing with the yesterday's topic, that is tools of recombinant DNA technology. I did discuss regarding the 6 different tools. Those were restriction enzymes, ligases, polymerases, as well as host and vector. So we will study about each of them in detail. So first one is restriction enzyme. So first restriction enzymes were discovered in such a way that there was E. coli which did not allow the growth of bacteriophage. Bacteriophage is a kind of virus which grows on bacteria. So E. coli did not allow the growth of bacteriophage on them due to presence of 2 enzymes. Out of these 2 enzymes, one added methyl group to DNA and the other acted as a restriction enzyme which did not allow the growth of bacteriophage. So first discovered restriction enzyme was Hind2. This restriction enzyme acts on a specific set or sequence of DNA. So there are almost 6 pairs of nucleotides in a DNA sequence which this Hind2 restriction enzyme did act on. That means what? This restriction enzymes are very much specific. They require specific DNA sequence so that they can show their activity. Now this 6 pair or a particular DNA sequence which is very much specific for this restriction enzyme, it is called recognition sequence. It is termed recognition sequence. Now Hind2 requires a specific DNA sequence which includes 6 pairs of nitrogen bases or nucleotides, which is termed recognition sequence. This first 6 pairs or these 6 pairs of DNA sequence were identified or isolated, but it was characterized only after 5 years. So that is about the first discovered restriction enzyme. Now how to name a restriction enzyme? For example, let us take an example of one restriction enzyme, that is E. coli. So the name for this particular restriction enzyme is given based on different rules. That is, the first letter represents the genus from which it has been extracted. The next 2 letters, it is about the species of the bacteria from which this particular restriction enzyme has been extracted, and R represents the strain. R represents the strain, whether it is a rough strain or a smooth strain. And last, that is Roman number 1, this represents the sequence, how it has been isolated from the bacteria. So this is how you name a particular restriction enzyme. When it comes to E. coli, it has been extracted from E. coli RY13 species. So E. coli RY13 is the one which has been the bacteria from which this E. coli restriction enzyme has been extracted. Now when it comes to this one, the first letter has been extracted from the genus, that is E, here it means Escherichia. Next two letters, that is coli. From there, that is the species name, the first two letters have been extracted. R represents the strain, whether it is a rough strain or a smooth strain. And Roman number 1 represents the sequence, how it has been isolated from the E. coli. So that is how you have to name a restriction enzyme. Next one, there are different types of restriction enzymes. Restriction enzymes, it belongs to a vast group of enzymes termed nucleases. Nucleases are the major group of enzymes to which restriction enzymes do belong. When it comes to the types of restriction enzymes, they are of two different types, that is exonucleases and endonucleases. If this is the DNA, this exonucleases enzymes acts on the ends of the DNA. DNA ends alli these exonucleases will be acting, which helps in cutting the ends of the DNA. Whereas endonuclease, if this is the DNA, they act within the DNA sequence. That means in between the DNA sequence also they can act and break the specific region. That is what is endonuclease, as the name suggests, endo and exo. Exonucleases restriction enzymes acts on the ends of the DNA, whereas endonucleases enzymes, they are very much specific, just like exonuclease, but they act within the DNA. Both of them are commonly called biological seizers because they help in breaking down or cutting down the DNA sequence. These restriction enzymes, they act on double stranded DNA. And here you already know that DNA are made up of sugar and phosphate backbone. So first, once the endonuclease or exonuclease act on a DNA, they break the sugar phosphate bond. So backbone of the DNA will be broken down. That is how they break into smaller DNA sequences or base pairs. They act upon, these sequences are normally palindromic sequence. Now what is a palindromic sequence anta keladre? For example, now this is a word Malayalam. Okay, this can be read from either sides on the same way. That means you can read it from right hand side to left hand side or vice versa. The meaning as well as the term remains the same thing. That is what is called a palindromic sequence. So what is a palindromic sequence in DNA if you ask? For example, there are 4 nitrogen bases. Now if you imagine this is the double stranded DNA GAATTC. So normally C pairs with G, T with A. So it will be the same thing. Now from 5 prime to 3 prime, the arrangement of the sequence will be GAATTC, whereas from the 5 prime to 3 prime, GAATTC is the arrangement. This type of sequence can be called palindromic sequence because if you read it from either sides, it remains the same. So normally this endonuclease or exonuclease, or in general the restriction enzymes, they act upon the palindromic sequence. Next we will have to study about action of restriction enzymes. Okay, this is the action of restriction enzymes. So here we have taken two DNA sequence. One is a vector DNA, another is a foreign DNA. Now I have elaborated or magnified the DNA in a sequence where it is having GAA-TTC. So this is a specific sequence which is necessary for E. coli restriction enzyme to act on. So here in both the vector as well as foreign DNA, I have enlarged the part where the sequence is GAA-TTC because the E. coli restriction enzymes acts only on G and A, that means between the guanine and adenosine nucleotides. This will be, or nitrogen bases, this particular restriction enzyme will be showing its action on. So when it comes to this particular sequence, E. coli acts between guanine as well as adenosine and breaks the sequence or nitrogen bases. Now here the enzyme cuts both the DNA at the same site, guanine and adenosine nitrogen bases. Next step is, you can see after the reaction or action of enzyme restriction, there are vector DNA and foreign DNA which has been put into separate parts, or they are separated. These ends, now if you see here the sequence is cut like this, where E. coli has acted on the upper sequence where AATTC is removed, only G is retained. Where in the foreign DNA you can see that the lower part only G is retained. So the next step is, you can see here the foreign as well as the vector DNA which is having cut parts. That means I told you that they act between guanine and adenosine. So in the upper part the AAA-TTC has been removed, whereas in the lower part the CTTA is retained and G is removed. Next, when it comes to the foreign DNA, here the G has been removed, AATTC is retained, whereas in the lower sequence G is retained, rest all is removed. Now these both ends are termed sticky ends. Why are these both ends termed sticky ends? Because whenever they come across their complementary counterpart, they start forming hydrogen bonds. ಈ sticky ಎಂಡ್ಸ್ಗಳು ಒಂದು ಸರಿ ತನ್ನ ಕಾಂಪ್ಲಿಮೆಂಟರಿ ಪಾರ್ಟ್ಸ್ ಜೊತೆಗೆ ಕಂಬೈನ್ ಆದ ತಕ್ಷಣ ಹೈಡ್ರೋಜನ್ ಬಾಂಡನ್ನು ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡೋದಕ್ಕೆ ಸ್ಟಾರ್ಟ್ ಮಾಡುತ್ತೆ. ಆದ್ದರಿಂದ ಇದನ್ನು, ಈ ಎಂಡ್ಸನ್ನು ಸ್ಟಿಕ್ಕಿ ಎಂಡ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ಸೊ ಒನ್ಸ್ ದಿಸ್ ಮಚ್ ಇಸ್ ಡನ್, the foreign DNA is incorporated into the vector DNA which helps in forming a recombinant DNA. Now this sequence, this cut part is joined with this particular sequence where the G is having a continuous AATTC, whereas the lower part CTTA will be joining with the G part which is retained in the foreign DNA. So in together they combine to form a recombinant DNA. That is how the action of restriction enzymes take place. Once again to discuss, the action of restriction enzyme is very much simple because we are using a particular enzyme which is acting on both the vector DNA as well as the foreign DNA. Here the restriction enzyme used is E. coli. This E. coli acts on a specific DNA sequence or recognition sequence. That sequence will be GAA-TTC, where the E. coli acts on G and A, that is between guanine and adenosine, the action of E. coli takes place. Now in the first step you can see that we have acted or we have applied the E. coli on both foreign and vector DNA, where the GAA sequence will be destroyed and the action of this E. coli will be between the guanine and adenosine. In the next step you can see both the foreign DNA as well as the vector DNA, both are having cut ends. These cut ends are termed sticky ends. Why are they called sticky ends? Because once they come across their complementary sequence, they start to produce the hydrogen bonds. That is why they are termed sticky ends. Next step is, these sticky ends should be joined together and that will be done to form a recombinant DNA. Now the foreign DNA is incorporated into the vector DNA, forming a recombinant DNA. This is how action of restriction enzymes takes place. Next we have to study steps involved in formation of a recombinant DNA using this restriction enzymes. here is the diagram which represents the recombinant DNA technology using the restriction enzymes. Now, here is a foreign DNA and here is a vector plasmid DNA. So, both will be applied with same type of restriction enzyme. If it is E. coli for foreign DNA, it will be the same restriction enzyme for vector plasmid DNA. Now, here we will be applying the restriction enzyme. So, they cut at a specific recognition sequence. Second step is, you can see here a specific sequence has been cut and the rest of the DNA has been separated. Same takes place even in the vector plasmid, where the vector plasmid has been applied with the same restriction enzyme and a small part has been cut. Next one is this particular DNA sequence which we have obtained from foreign DNA should be incorporated into this particular vector. That process is done by an enzyme called ligase. Ligase acts on the cut ends of the DNA. That is why here ligase enzyme is used to incorporate this foreign DNA into this vector DNA. Once that incorporation is successful, this particular plasmid is termed recombinant DNA because it has a DNA sequence which we have obtained from the vector and a DNA sequence which is obtained from the foreign DNA. That is what is called a recombinant DNA. Now, this recombinant DNA is introduced into the host through a process of transformation, and here the host is E. coli. So, this recombinant DNA is introduced into the E. coli. This bacteria, when it multiplies itself or undergoes reproduction, they produce daughter cells, and these daughter cells also have this plasmid which have been replicated using DNA polymerase enzyme. So, this is the diagrammatic representation for recombinant DNA technology. That is about the steps involved in recombinant DNA technology using restriction enzymes as well as diagrammatic representation for recombinant DNA technology. Next, we will have to study about, once the DNA sequences are there with us, how we can separate them, that is isolation or separation of DNA. So, separation and isolation of DNA takes place with the help of a setup which is termed gel electrophoresis. So, the cut strands of DNA. Now, how are these DNA strands cut? These are cut because of presence of restriction enzymes. Once the restriction enzymes act on the DNA, they are cut into smaller pieces. This cut DNA is introduced into this box-like structure which are termed wells. So once the cut DNA strands are introduced into the wells, normally DNAs are negatively charged. So in gel electrophoresis, what we do is we apply this with the electric field and make the DNA move towards the anode. That is, the negative DNA should move towards the positive charge using a electric field. DNA ಅನ್ನು ಸೆಪರೇಟ್ ಅಥವಾ ಐಸೋಲೇಟ್ ಮಾಡುವಂಥದ್ದು ಹೇಗೆ ಅಂತ ಹೇಳಿದ್ರೆ, DNA ಸ್ಟ್ಯಾಂಡ್ಸ್ ಮೇಲೆ ರಿಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಜೈಮ್ಸ್ಗಳು ಆಕ್ಟ್ ಆಗ್ತವೆ. ಈ ರಿಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಜೈಮ್ಸ್ಗಳು ಲಾಂಗ್ ಸ್ಟ್ಯಾಂಡ್ಸ್ ಆಫ್ DNA ಅನ್ನು ಸ್ಮಾಲರ್ ಸ್ಟ್ಯಾಂಡ್ಸ್ ಅಥವಾ ಸ್ಮಾಲರ್ ಪೀಸಸ್ ಆಗಿ ಸೆಪರೇಟ್ ಮಾಡ್ತವೆ. ಸೊ ಈ ಸ್ಮಾಲರ್ ಪೀಸಸ್ ಅನ್ನು ನಾವು ಇಲ್ಲಿರುವಂತಹ ಬಾಕ್ಸಸ್ ಅದನ್ನು ವೆಲ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ಈ ವೆಲ್ಸ್ಗಳಲ್ಲಿ ಲೋಡ್ ಮಾಡ್ಕೋತೀವಿ. DNA ನೇಚರ್ ವೈಸ್ ಆರ್ ನ್ಯಾಚುರಲಿ ನೆಗೆಟಿವ್ಲಿ ಚಾರ್ಜ್ ಆಗಿರುತ್ತೆ. ಸೊ ಈ ಜೆಲ್ ಎಲೆಕ್ಟ್ರೋಪೋರೋಸಿಸ್ ಅಲ್ಲಿ ನಾವು ಆನೋಡ್ ಆಕ್ಷನ್ ಇಂದ ಈ ನೆಗೆಟಿವ್ DNAs ಅನ್ನು ಪಾಸಿಟಿವ್ ಎಲೆಕ್ಟ್ರಿಕ್ ಫೀಲ್ಡ್ ಹತ್ರಕ್ಕೆ ಮೂವ್ ಮಾಡುವಂಥದ್ದು. ಸೊ ಹಿಯರ್ ಒನ್ಸ್ ದೇ ಆರ್ ಲೋಡೆಡ್ ಇನ್ ದ ವೆಲ್, the electric field is applied on this gel electrophoresis. The DNAs move towards the anode which is positively charged. So if the DNA should move towards the positive charge, they should have some matrix here. This matrix will be agarose gel. Agarose gel is a natural polymer which is extracted from seaweeds. So here what happens is, the gel electrophoresis contains wells to load the DNA and electric field is applied so that the DNA move towards the anode. In the gel electrophoresis, it contains a matrix so that it helps the DNA to move easily. The gel which is present here, or the matrix which is present here, is a agarose gel. This agarose gel is a natural polymer which is extracted from the seaweed. If you see here, the larger DNA strands are much more close towards the well, whereas the thinner or smaller strands move fast towards the anode. The thinner or smaller DNA strands are very close towards the anode, that means they are moving fast compared to the thicker strands. Thicker strands will be much more close towards the well region. Next, once the DNA is transferred here, it should be stained. If it should be observed, then it should be stained. The staining is done with the help of ethidium bromide. Ethidium bromide is used as a stain to identify the DNAs in the agarose gel. If it is not stained, it doesn't appear in the, or it is not projected in the agarose gel. That is why ethidium bromide is added into the gel electrophoresis, where you can see this strands, but it is not observed under visible light, but you have to use UV rays to observe this DNA strands. When UV rays are focused on this gel electrophoresis, the stained DNA strands appear to be orange in color. ಈ DNA strands ಅನ್ನು ವಿಸಿಬಲ್ ಲೈಟ್ ಅಲ್ಲಿ ಒಬ್ಸರ್ವ್ ಮಾಡೋದಕ್ಕೆ ಸಾಧ್ಯ ಇಲ್ಲ. ಅದಕ್ಕೋಸ್ಕರ ಇದನ್ನು ಇನಿಷಿಯಲ್ ಆಗಿ ಸ್ಟೇನ್ ಮಾಡಬೇಕು. ಈ ಸ್ಟೇನಿಂಗ್ ಪ್ರೋಸೆಸ್ ಹೇಗಾಗುತ್ತೆ? ವಿತ್ ದ ಹೆಲ್ಪ್ ಆಫ್ ethidium bromide. Even though you add ethidium bromide to the DNA, it cannot be visible because the visible light fails to project the DNA in the agarose gel. So that is why you have to use UV rays. While you use UV rays which is projected on this gel electrophoresis, you can see that DNA strand appears to be orange in color. Where the DNAs are not there, it appears to be just transparent. That is why ethidium bromide is used to stain and the observation to be done in UV rays. Once you observe the DNA, now you have to separate this DNA from this gel electrophoresis. Part by part, pieces by pieces, the DNA can be separated from this gel electrophoresis by a method called elution. Elution is a process from which you can separate the strands of DNA from the gel electrophoresis. Once this elution process is done, next the pieces of DNA will be transferred into the vector plasmid to to form the recombinant DNA. After the elution process, this particular DNA strand is transferred into the vector plasmid, or you can call them cloning vectors, to form the recombinant DNA. That is why separation and isolation of DNA is very much necessary in recombinant DNA technology. So here what we are doing, the restriction enzymes acts on the DNA and cuts the DNA into smaller pieces. Now here in this gel electrophoresis, what is to be done? The double stranded pieces of DNA should be separated. So initially they are loaded into the wells, and as they are negatively charged, electric field is applied on this gel electrophoresis so that the DNA strands move towards the anode. Once the well is loaded with the DNA, electric field is applied, as well as agarose gel is present, which helps in easy movement of the DNA towards the anode. This agarose gel is a natural polymer which is extracted from the seaweed. Once electric field is applied, the larger strands stay closer towards the well and the smaller and thinner strands move faster towards the anode. Once this process is done, next it should be separated. So to separate, we should know where exactly is the DNA. For this purpose, ethidium bromide is the stain which stains the DNA, and when it is observed under UV rays, the DNA strands appear to be orange in color. Once the orange color DNA is easily visible in this gel electrophoresis, they can be separated from this gel using a process called elution. Once elution process is completed, this DNA strands are now ready to incorporate within the cloning vectors to form a recombinant DNA. So that is all about the separation and isolation of DNA. So we will end the session here. In today's class, we did discuss about the first tool of recombinant DNA technology, that is restriction enzymes. We did learn how different steps are involved in the restriction enzyme to form a recombinant DNA technology, as well as we have also discussed the diagrammatic representation of recombinant DNA technology, and at last we did discuss regarding the separation and isolation of DNA using a process of gel electrophoresis. So that is all in today's session. In tomorrow's session, we will be learning about cloning vectors as well as host. Thank you.